For meiosis class, as what I have told you, meiosis is observed among gametes or sperm cell and egg cell formation. We call that as gametogenesis. Genesis means formation. Gameto refers to gametes. Okay? Now, unlike mitosis, meiosis is said to be longer. Why? Because it is divided into two. We have meiosis 1 and meiosis 2. So this one is meiosis 1 and this one is another division, meiosis 2. Okay? Now let's start. Now meiosis 1 is divided again into 4 sub-stages. You have prophase 1, metaphase 1, anaphase 1, and of course telophase 1. Okay? Now here, the longest period in meiosis is prophase. This is said to be, or prophase 1. That's the longest period. Why? Now to start with, the same thing happened. The sperm, or say, where is the site for the sperm cell production? It's in the testes. For the egg cell production is in the ovaries. So there are 2 ovaries, the left and right. And every 28-day cycle, there would only be 1 egg that is being produced. However, for the sperm cell production in the testes, or the one that is hanging in the testicles, I mean in the scrotum, I should say. Now, that produces millions of sperm cells, or say the sperm cells dividing in the testes. Now, here it starts with prophase 1. Now, as what I said, it is said to be the longest period because it is said to be divided again into 5 sub-stages. Okay? So, you have here, and what is the most important event in prophase 1? Now, what makes it longer? It's because of this important event. 2 important events, we call that as synapsis, or synapse formation, or the pairing of homologous chromosomes. When you say homologous class, paris, they have partners, homologous. Now, in mitosis, there is no homologous, no partner in mitosis. But in the sperm and egg formation, the chromosomes have their own pair. Okay? They have their own pair. So that's why, kani siya iyang paris, kani po iyang, you have here paris. So we have the X and the Y later, I will talk about it. Okay, so you have here the partner. Partner ni sila. Kaning pink, partner na sila, then kaning blue. So actually, it's not blue or pink. It's just labeled as such that will indicate that they are said to be homologous or partner. Okay? So kanang partner, maunang mag-uban permi. Maunang mag-uban permi. So here, at prophase 1, you could see here the partner. There is already synapse formation at prophase 1. And kaning partner, prophase 1, kanang partner, they will cross over. How is crossing over? What doing, or say, how is it be done? If this is the double-stranded chromosome, the homologous chromosome, here, say, there is what we call as overlapping, or we term that as crossing over. So this is the most important event in the meiosis. Crossing over, we call that cross over. Now what happened in the crossover? So, kanina homologous chromosome overlap siya dito sa other partner niya. No? So there is, now during this time, the crossing over, there is exchange of genetic materials. Give and take na sila, or say, mag-share. Mag-share na sila. So, kaning nag-overlap diri, kaning nga area, kaning nga materials, matanggal. That will be dissociated, kana siya nga segment, then matransfer dito sa PCAS. And then, kanina pong na-overlap sa PCAS, matransfer na po diri sa isa ka homolog, no, sa iyang partner. So there is what we call as exchange of genetic material. As you could see here, na no? Nag-crossover ni sila. Kani sila mo ay partner. Kani mo ay partner pod. Homologous chromosomes, pareho sila size usually. No, pareho size. No? And ang katong homologous chromosomes mao toy mag-crossover. Mao toy mag-crossover. Now, you have noticed nga ang portion aning isa na diri. Ang portion ko diri na diri. There is what we call as exchange of genetic materials. Now, kani siyang event, this is very, very important. Remember, paghibo ni sa sperm ug egg. Kani crossing over, this is very, very critical. Should there be any factors that will disturb this, then this will contribute to the abnormalities of the sperm or egg. Now, what if ang abnormal sperm no ay naka-fertilize sa egg? That will result to abnormal individuals. Or ang egg na abnormal, mo ay na-fertilize sa sperm. So, that will still result to abnormal individuals. So, this is very, very critical. So, dapat dili siya i-disturb during this time. So, there is what we call as exchange of genetic material during crossing over. And this occurs during, this occurs during prophase 1. The paris-paris or the synapse formation is in prophase 1 and the crossing over is still in prophase 1. Now, at metaphase 1, katong homologous chromosomes nga nag-crossover, they line up on the equatorial plate. They line up on the equatorial plate pero na kay mga indicators na they have exchanged already. Nag-exchange na sila. Now, at anaphase, so remember, they have to be attached to the spindle. Still, pare-pare po sa meiosis. They need to be attached to the spindle and then they line up on the equatorial plate at metaphase 1, but duha-duha sila. Oh, the homologous chromosomes line up on the equatorial plate. However, during anaphase 1, ang nagparis-paris, no, the homologous chromosomes start to separate. Pag-separate nila, still double-stranded. Kani double-stranded pod. Pero say, if this is 46 class, pag-separate ani karon 23 chromosomes but still double-stranded. So, duha ka strands. Kani sister chromatid, kani sister chromatid. Sister chromatid ani, sister chromatid ana. Still double-stranded. But the homologous chromosomes separate. Nagbaybay na sila sa ilang partner. Okay? And of course, you could see also groove or furrow for the animal cells of sperm or egg man. No? So, groove or furrow which starts the cytokinesis. Okay? So, the cell, after anaphase 1, it starts to form groove or furrow. Pag telophase 1, there is now separation of these two cells. So, there are two cells formed. Pila ka chromosomes? 23, but double-stranded. And after meiosis 1, the cell will not proceed right away to meiosis 2. There is what we call as interkinesis. There is interkinesis. Now, what happened here? Interkinesis is the gap between meiosis 1 and meiosis 2. Now, it's similar with interphase period. However, so at that time, interkinesis, ang katong duha, these two will, during interkinesis, mo grow. So, there is growth. Mura siya interphase period, pero there is no S phase. Naman siya growth. Mo grow lang siya. At interkinesis. This is the gap between meiosis 1 and meiosis 2. Okay. So, after interkinesis, after that gap, then the cell would start again to divide for meiosis 2. Now, meiosis 2 is subdivided again into 4 substages. We have prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, remember, ang result sa telophase 1 are 2 cells, duha ka cells. You have 2 cells, 1 and 2. But they are no longer homologous. Meaning, ang partner nag-separate na. They said goodbye to each other. Okay? Now, here. Now, at prophase 2, mag-start na po mura na siya meiosis. No? Pero duha ka cells sila nag-divide. Duha ka cells nag-divide. So, at metaphase 2, there is again lining up of chromosomes. This time, double-stranded siya, pero wala na yung homologous chromosomes. Kaya nga nag-separate na sila into one cell. Hilain na sila balay. Separate na ilang balay. 2 cells are dividing. Okay? So, there are double-stranded chromosomes. Now, remember, if they start with 46, kakaroon 23 each. Kani 23. Now, they line up at the equatorial plane. Chromosomes line up at the equatorial plane. And anaphase 2, diri sa anaphase 2 na pod, it's the sister chromatids separate. So, kani siya, nag-divide na kani siya. Kani, nag-divide. So, nanaputay groove or furrow. So, kaning isa ka cell mo divide into 2, and then ang ilang chromosomes na separate na po. So, kaning isa ka chromatid ni ato sa left, ang isa ka chromatid ni ato sa right. The same is true with the other chromatids. So, na po'y separation. So, ang duha ka cells to start with, nahimo na 4 cells. So, this is in anaphase 2. In anaphase 2, there is what we call a separation of sister chromatids. At telophase 2, you could see here how many cells? There are 4 cells. 1, 2, 3, 4. But these 4 cells class are said to be different from each other. Unlike in meiosis, where the 2 cells are exactly the same. Pareho sila exactly the same, similar with each other. But here, in meiosis, the 4 cells formed are exactly different from each other. Lahi-lahi ilang mga chromosomes. Now, that explains why, say, if there are 12 children in a family with the same mother and father, except for identical twins, they are all different from each other. What causes such? The egg that is produced by your mother every month is exactly different from the previous month. Okay? And it will be different for the next month. That is said to be unique because of crossing over. And of course, sa inyong sperm cell, the sperm cell produced by your father, for every 72 hours, there are millions, would be different from each other because of crossing over. And it would be different for the next set of spermatogenesis. They are exactly different from each other. So, ang produkto sa mama o papa, nga sperm o egg, are different from each other. So, every pregnancy, lahi o dagway ang sperm o ang egg. And that explains why, say, 24 mo kabuok in a family with one mother and father, lahi-lahi mo dagway. What causes such differences? because of, or we call that variation. What causes such? Because of crossing over. Okay? Now, you know what? Crossing over is very important. Now, what if, halimbawa, na ay abnormal nga kaliwat imong papa or imong mama, say, halimbawa na inherit yun niya ang katong abnormal nga sperm or it could be abnormal nga egg. Now, kung walay variation, walay crossing over, kamong tanan mga anak maka-inherit atong mga abnormalities nga nasa sperm or nasa egg. So, God intends that there would be crossing over so that the sperm and the egg that are, the sperm cells and the egg that are produced, every time there is meiosis or spermatogenesis and oogenesis would be different. Okay? Now, for, say, the production of sperm cells, we term that as spermatogenesis. Yes, spermatogenesis. Genesis means formation. Then, sperma refers to sperm cells. No? Now, where is it from? Okay, so the overall you have that. Meiosis 1, meiosis 2. Those are mga results of abnormalities. No? Mga abnormalities na siya. Okay. Now, here in the sperm cell production, where is it formed? Where does it occur? It occurs in the testes. So every 72 hours, there are millions of sperm cells that are or mga millions of sperm cells that are destined to undergo spermatogenesis. So here we have the sac. We call that sac. Sac, or say you have spermatogonium, singular, plural spermatogonia. Now there are so many. The testes consists of several spermatogonia, plural, and one of the cells there will grow and differentiate into primary spermatocyte. Ang primary spermatocyte, which is 46, 46 ka buok, diploid, 2 sets, mo undergo og meiosis 1. So mahimo ng 23 diri, 23 pod. This is the summary, or summary sa meiosis. Now, this is how spermatogenesis is done. This is the diagram. Now, the one that will undergo meiosis 1 is the primary spermatocyte. Muna siya mo undergo og meiosis 1. Ang resulta sa meiosis 1, ang primary spermatocyte will be converted into secondary spermatocyte. So there will be 2. So kani siya So, kani siya mao na ni ang kana siya mao ang primary spermatocyte. Mo ni primary spermatocyte mo undergo meiosis 1. Ang resulta mo produce siya og secondary spermatocytes. Kani mao na ning duha. And then mo undergo na kuni siya og division kaning secondary spermatocyte para mo produce og 4 haploid. We call that haploid. 23 chromosomes na lang. 1 set. So, ang naa nga chromosome sa sperm or sa egg is 1 set lang. So, this is 1 set. So, kani siya, mao na ni ang mga spermatids. Upat ka buok sa isa ka cell. Sa isa ka primary spermatocyte. So, that results to the production of 4 spermatids. Now, kaning spermatids class, dili pa na maka-fertilize sa egg. It has to undergo growth. and differentiation until mahimo na siyang spermatozoa or sperm cells. So meaning, nag-differentiate na siya, na siya ikog. So kung nanay ikog, matured na siya, pwede na siya maka-swim toward the egg. Okay? So, mo nining maka-fertilize. Okay? Now, for the egg, the oogenesis, we term that oogenesis. Oo means ovum, genesis means formation. This occurs in the ovaries. Now, every 28 day, no, kaisa lang ka sa isa ka bulan, kaisa lang normally, kaisa lang oogenesis. Ang sa lalaki, every 72 hours mag-spermatogenesis. And there are millions of primary spermatocytes that will undergo meiosis 1 and 2. But for the oogenesis, there will be one, say, in a sac, no, sa ovaries, there are so many sacs there, mga hundreds lang, kana millions. Kani hundreds of oogonia, plural, singular oogonium. One of the cells there will grow and differentiate into primary oocyte. And the primary oocyte will undergo meiosis 1, and that will result to unequal, unfair division of the cytoplasm. Unfair iyang cytokinesis. Ang isa gamay, ang iyang cytoplasm, ang isa dako. No? Dako. Now, ang gamay, ang first na gamay na cytoplasm, we call that first polar body. Now, this first polar body may or may not undergo meiosis 2. So, para mahimo na po siyang second polar bodies. But, for this secondary oocyte, so, kanang nakaresibog dakong cytokinesis, or cytoplasm, I should say, mo undergo siya og meiosis 2. Okay? And, naana po'y unfairness. One receives greater cytoplasmic materials than the other. Ang katungga ay gamay ra kay cytoplasm is what that forms into polar body. We call that the second polar body. The one that receives more of the cytoplasmic material or materials is now formed into oocyte. So, unlike the spermatogenesis, ang spermatogenesis after meiosis 2 or telophase 2, upat ka buok functional, later on, 4 cells form. 4 haploid cells form. But in oogenesis, there is only 1 haploid cell form. We call that oocyte. And this oocyte will grow and differentiate into ovum, or we call that as egg cell. So every month or every 28-day cycle, you are only the female is producing 1 functional egg. Now, that is normal. However, there are some abnormalities. Usahay maglayag. Usahay walay menstruation. Kaya sa halimbawa, kaning egg, say, it will just stay maybe mga nanay egg, no? In the ovary. Later on, during ovulation, that egg will be ruptured in a follicle. I-release siya sa fallopian tube. Magulat siya dito sa fallopian tube for 2 to 3 days. Kung walay Prince Charming, so it will just be degraded. And that will, together with the tissues formed in the uterus, so of course there are mga tissues formed in preparation for pregnancy. So mag-prepare ang uterus, ang imuhang womb, during ovulation period, should there be any fertilization. But kung walay sperm cell nga ma-deposit, tapos ana sa mga dalaga pa, walay sperm cell, ang egg cell will just be degraded. Mo-degrade lang siya in the fallopian tube, mga around 2 to 3 days for waiting, and then they will just go with the flow. No? So together with all those tissues formed in the uterus, and that becomes the mens or menstruation. Okay? So that is how wonderfully we are made. Marvelous are thy works. Okay? So